So, welcome everyone. We're gonna have this as a flipped classroom, which means that you're gonna have the lecture here now on videos, and during the class we're gonna work on it. And why? Because simply this is quite, let's say, tough, quite hard concepts to grasp. So I thought that, okay, maybe it's better we do it this way, so that way we can really work with these concepts. So today we're gonna then talk about how we identify selection in populations in genomes, and which is what we call adaptomics actually. Because surely one good thing that came with the genomic era is that we can really do selections or detect selection, let's say, in the whole genome. We can do it at the SNP level, we can do it at the gene level, we can do it at the whole genome level. So the genomic era has really been real progress in adaptomics. So let's do it. So you might remember this from your population genetics classes. This is what we call in French a la palissade, which means basically that if everything stays the same, nothing changes. And what does that mean? That simply means that if all these criteria that you have here are respected, then the allele frequencies in the population stay the same, okay? So no selection, no mutation, no migration, large enough populations, and random mating. Of course, this never happens, but that's precisely what we're testing. That's in population genetics, that's our null hypothesis. And we are precisely interested in knowing when one of these criteria is not respected and how this can explain the change of allele frequency in a population. And especially today, adaptomics. So we're especially interested to know when selection is acting, what's going to happen. But before we jump into selection, explaining everything, you have to know, or you have to be reminded of a very important paradigm, which is that basically most of the variation we see at the molecular level is not affecting fitness. It's not due to selection. So most of the things you're going to observe when you screen a genome, all the genetic variation, allele frequencies, whatever is all about something else, but not selection. And what is something else? Well, it's basically two main things: stochasticity, so pure luck, pure chance, pure randomness, and demographic processes. Like typically, if you see differences in allele frequencies or differences in sequences between two lineages, these differences are mostly not about selection. These differences are simply due to new mutations arising across time, and of course these mutations will by chance simply be different in two different lineages, or these two different lineages had a different demography history, such as bottlenecks we're going to talk about, colonization, etc., etc. So most of the molecular genetic variation we see in the wild has nothing to do with selection, and you have really to keep this in mind very importantly, because basically when we're going to search for selection, we're going to keep in mind this as a null hypothesis, which again is whatever allele frequencies, genetic variation is mostly about neutral evolution. So neutral evolution meaning no effect on the fitness, no selection. And if we can prove or test that something deviates from this null expectation, then we can consider that selection might be acting, but only then. And surely I was saying that genomics era really improved a lot what we know about selection, etc. But most of the things that we're going to talk today, and most of the things that are done in population genomics, are something extremely simple. We're measuring allele frequencies. That's all we do. And based on this allele frequency, we can infer what's going on, again, in a gene, in the whole genome, in the non-coding part of the genome, etc., etc. So the data we work with in population genomics is extremely simple. After all, it's just measuring allele frequencies and from that deducing what's happening. Of course, the genomics era allowed us to really increase the accuracy and the fineness we can have. We can work with a single SNP. We can measure the allele frequency of a single SNP. We can measure the allele frequency of the whole haplotype in a gene, the whole haplotype in a chromosome, in a genome, etc., etc. But keep in mind that most of the thing I'm going to show you is just about allele frequencies, nothing else. And this is what we're going to have. So this will be divided in several videos so that it's a bit more digestible. And that way you don't have to watch a whole lecture in one go. And that's much easier, I think, for you to handle. So, but let's start by reminding you a bit of what can happen in terms of demography and because again, as I said, demography is the most and stochasticity are the two main factors explaining genetic variation. So we need to go back to what these are. So of course, this is typical population genetics. You might have a bottleneck, which means reduction of the population size. And once a bottleneck happened, it can be because of a catastrophe or colonization event, etc., etc. After that, we're going to start to have recovery of this population. Recovery meaning that here genetic diversity and allele frequency will dramatically change. And with recovery, then we're going to have increasing individuals, but then with new mutations, we're going to recover also the genetic diversity and recover stable allele frequencies. And yeah, founder effect is a typical case of bottleneck where basically from a whole population, we're going to have a small group that will migrate. And of course, these specific individuals cannot carry all the genetic diversity of the original population, especially rare alleles like this triangle here, as just by chance, has a very low chance to be carried out in the new population that are colonizing a new environment, for example. So basically, a bottleneck is often associated with reduced genetic diversity, again, because by chance, not all the alleles can be maintained or can be transmitted to this tiny population. And because then what will happen is when there are very few individuals, that means that these individuals will go through inbreeding, and that will increase homozygosity, at least on the short term. And as I said, rare alleles, just by chance, have much less chance to be transmitted in this tiny population. So usually the rare alleles are much less frequent, and only the major alleles are maintained in these tiny populations. Another case which we're going to talk about later is of bottleneck is usually the loss of self-incompatibility in plants or transition to parthenogenesis in animals. So what's happening there is basically most of the plant species are self-incompatible. They cannot mate with themselves. They need another mate to be able to produce seeds to reproduce. But there is, like recurrently in plant evolution, there is a loss of this self-incompatibility. So basically it will promote selfing. So selfing meaning reproduction with oneself. And that is acting as a strong bottleneck. Why? Because simply, usually this mutation, this loss of self-incompatibility happens in a very few number of individuals. And these new few individuals will then create a selfing lineage. So that creates by default a strong bottleneck. Okay, and there is one first measurement, one measure that we need or that we often use in population genetics, and you probably know it, it's called the effective population size. What does that mean? That means that this effective population size is the size of an ideal population, no inbreeding, etc., which means that with this population that is, let's say, healthy in terms of genetics, we need certain size to recover the total number of alleles of the population we are looking at. That means what? That means that usually the effective population size is actually much smaller than the real population, because the actual real population is very often ongoing to a certain extent to some inbreeding. It's not the most variable that it could be. So that means that usually we could reduce this population to a very few number of individuals that would be sort of perfect, carrying all the diversity that is present in this population. And for example, this effective population size decreases, of course, with a bottleneck, but also typically here, another factor that will decrease the effective population size is the sex ratio. Simple way to put it is, if you look here, you see that for a sex ratio which is 1 to 100, then while the actual population remains 100 individuals, the effective population size decreased to close to 0, and it's actually 1. And the reason why is because you have to imagine that every generation on the, let's say, male side, for example, there is one single individual contributing to the next generation. So that means that with one individual, we are able to basically recover the whole genetic diversity of the next generation, because again, all the next generation had only one single, let's say, male as a parent. And of course, the next generation, if the sex ratio is maintained, it's exactly the same, and it's getting worse and worse because even all the females inherit alleles from one single individual that was a male. So both male and female have one single parent. So that will decrease the effective population size. And typically in humans, the estimated effective population size is about 10,000. Again, that means what? That means that out of the billions we are on Earth, we would need only about 10,000 humans to catch the whole genetic diversity that exists in the whole humankind. And that's very simple to explain. We all know that there were a really strong bottleneck, like migration of very few individuals from Africa that colonize the whole Earth. So of course, it's easy to imagine that indeed we are extremely inbred, and again, a very few individuals would be enough to recover the whole genetic diversity of humankind. So this is a very important factor in population genetics, and we will see why. And we're gonna see now, and imagine that actually we start with a population that is relatively healthy, so kind of large effective population size, so high genetic diversity, etc., etc. In this population, actually, selection will act very efficiently. Why? Because the only thing that probably will change the frequencies of alleles is going to be about how deleterious or how advantageous it is. Then if it's advantageous, it will spread to the population, like, for example, the purple one here will spread to the population. And on the other hand, stochasticity, so drift, which means that change of allele frequency by chance is very unlikely to happen. Why? Because there are enough individuals to buffer it. So even if by chance one individual dies, that has nothing to do with genetics, but simply chance, there are other individuals carrying the same allele. So frequencies will not change too much due to drift, to stochasticity. But now imagine that we have a different case. We actually have reduction in the real population size, but also in the effective population size, because you see that there are very few alleles left, okay, in this population. There is much more probability that actually one allele will get fixed by pure stochasticity, by pure chance. While selection here will have much harder time to act. And typically it takes the case, okay, for whatever reason, the blue allele will disappear from the population because there is just one individual carrying it. So if this individual fall from a cliff, for example, then it's over. This allele is out of this population. So in this case, surely we will have fixation of an advantageous allele, but not due to selection, simply due to stochasticity. But we can imagine the exact opposite, that the purple allele is carried by one individual that will fall from a cliff, and then we are left with the deleterious allele in the population. So for this reason, then you can see that indeed, when we have low like actual but also low effective population size, and selection has much harder time to act. And instead, drift and stochasticity will have a much stronger effect on allele frequencies. So in general, low effective population size, low efficacy of selection. Like selection has hard time to act. And let's continue with this, which is completely linked. So imagine now that there is these two individuals, okay? So these two individuals have whatever, one chromosome and they're diploid, so they have two chromosomes, each of them. You have now on this chromosome 3 loci, A, B, C, and then you see that there are different alleles, prime, double prime, whatever, plus, etc., etc. The important thing is basically here, A and B are linked. They are what we call, they are in linkage disequilibrium. What does that mean to be in linkage disequilibrium for two loci? That simply means that these two loci, the alleles at these two loci will always be inherited together in the next generation. And vice versa, if there is no or very weak linkage disequilibrium between two loci, that means that the alleles in these two loci will never be inherited together. And for example, this is a case here where A and B are linked. They are in linkage disequilibrium because you see that when we find prime in A, we always find prime in B. And on the individual number 2, when you have A plus, you also have B plus. But this is different when you look at the loci C, because in this case, you cannot see really a link between, you cannot predict the allele in A when you know the allele in C. So basically, the alleles are inherited independently in C versus the A and B. And the reason for that is pretty simple. It's simply you have recombination between them. And because you have recombination between C in one hand and AB on the other hand, then thanks to recombination, as you know how it works, then allele A prime can be recombined, and then you can have C double prime, or you can have C plus or C prime, whatever in the new chromosome being formed after recombination. But on the other hand, there is no recombination happening between A and B. That means that A prime and B prime typically will always be inherited together after meiosis in the next generation. So this idea of linkage disequilibrium is really about what is the probability that two loci segregate together. Segregates together means being inherited together after meiosis happened and to the next generation. Okay, so surely linkage disequilibrium, what is like the probability that two alleles are inherited together depends on how much recombination happens, of course. If there is a lot of recombination happening between two loci, again, these two loci have no chance to ever be inherited together. I mean, the alleles as these two loci have no chance to be inherited together because there's going to be a lot of crossover and no possible link, genetic link between them. But actually, linkage disequilibrium, we use it often in population genetics as a marker for what we called effective recombination. So what effective recombination means, that simply means what's going to be the power of recombination to mix alleles together, okay? Or not precisely not together, but rather to keep alleles independent from each other in terms of inheritance. And again, as I said, if there is recombinations between two loci, of course, the alleles at these loci will effectively be recombined, okay? So there will be a lot of very strong, very effective recombination between these two loci. And that's how it would look like. So imagine that you have two loci that are very far from each other. The further they are from each other, the more chance there is to have recombination hotspot between them. So that means that the probability of recombination between them is really high if they are very far from each other. If there are two sides of the chromosome, they are very far from each other, so there's going to be a lot of recombination happening between them. And that means what? That means that the linkage disequilibrium between them is going to be super, super low. The probability that the alleles will be inherited together in these two loci is close to 0. So one is actually the mirror of the other, like the negative mirror. Okay, so of course, how much recombination events happen is really important factor for effective recombination. Now there is another really important factor for effective recombination to happen. And that important factor is what we call, is homozygosity. So imagine that you have the case again here as before, except that this time this individual is purely homozygote for the whole 3 loci. Here, recombination is going to be not very effective at all. Why? We still have recombination happening between B and C. The recombination are spotted there, but the thing is, because C plus and A prime, B prime, A prime, B prime, all these 3 loci are homozygotes. That means what? That means that it doesn't matter that there is recombination or not, because B prime will always be found with C plus, and A prime will always be found with C plus, and A prime with B prime, but that we knew already because there is no recombination happening between them. So that means what? That means with high homozygosity, we're gonna have very low, very inefficient recombination. Because even if physically recombination happens, it's not gonna help anything to mix alleles or to create a lot of different allele combinations, because there is simply no different alleles to make different combinations. Everything is homozygote, so even if C plus is recombined with this other chromosome, which also has C plus, nothing will change, okay? Like the recombination will simply be useless. And you might wonder why I'm telling you this. Well, now imagine something. Imagine that we're going back to this idea of effective population size, effective recombination, and efficacy of selection. So same case here, okay? So individual, which is homozygote. But now let's add another information, which is that each of these 3 alleles have a different effect on the fitness. The A prime allele is advantageous, B prime is neutral, so no effect, and C plus is actually deleterious. You might see what's coming here. The problem that we have here is that selection is going to be very inefficient. Why? Because ideally, to have a highly high fit individual would mean having an individual carrying the allele A prime, but not carrying the allele C plus. Okay, so only carrying the advantageous allele, but not the deleterious one. But in this case, it's simply impossible. It's simply impossible. Why? Again, because there is no possible recombination that could disentangle or disconnect the A prime from C plus. Okay, so even if there's gonna be positive selection on the A prime happening, there at the same time will be negative selection on the C plus. But because they are linked genetically, because there is no effective recombination between them, that means that always A prime will be selected together with C plus. And that means that C plus selection will not be able to purge out this C plus allele because it's linked with the beneficial allele. And that's what we call allele hitchhiking, which precisely means that the C plus is hitchhiking the A prime allele. So it's, even if it's deleterious, it's gonna be maintained in the population because it's linked with an allele which is advantageous. The selection cannot purge out deleterious allele and at the same time spreading the advantageous allele in the population because they are linked together. So effective recombination is extremely low here. And if we continue with this idea of low and homozygosity and inefficient selection. So of course, homozygosity will increase with specific events such as selfing, as I told you before. So transition to selfing is going to increase homozygosity, sorry. And same with inbreeding. So very few individuals that will generate a new population. They're going to be a very, very strong inbreeding, and that will also lead to homozygosity. So let's make the calculation here. So imagine that you have, to start with, one single individual, which is heterozygote for one locus. So a big A, small a. And it will then, as I said, it carries a mutation that allows him or her, whatever, to be a selfer. So it will be able to reproduce with itself to create a new lineage. So I'm asking you, what will be the genotype of the F1 generation and same F2 generation? So from the F1, each F1 will have the same capacity to self. That means that each F1 will self with itself and create a new generation, and this new generation will be then the F2. So do it like I'm asking you to pause this video and to think about it, to make a Mendel table, that's called Mendel segregation table, with this capital A, small a, and tell me when it's selfing, what are the proportion of heterozygote individual, homozygote individual, etc., etc. And once you've done that with F1, you do the same with F2. So pause this video and then we start again. Okay, so you might have found this. So this is classical Mendel table, and from the individual which is heterozygote, we have two possible gametes on the male side, which is capital A or small a, and exact same thing here for the female gamete, big A, small a. That gives what? That gives that out of this, 50% of the individuals will be heterozygote and 50% will be homozygote. So that means what? That means that from a population, one individual being 100% heterozygote, we already lose half of it. So half of the individuals will be heterozygotes. Now, let's do the same. So for the F2s, that was for the F1. So for the F2s, it's the exact same thing. So obviously, the individuals that are homozygotes, big A, and small a, will only be able to give one genotype, which is big A for the big A, and small a. Okay, so these 50% that are homozygotes, they will not become heterozygotes again. And of course, unless we consider new mutations, but that's much slower than regeneration happening. So that means that there are only left the other 50% that are heterozygotes that can create or generate different genotypes. And basically, it's the exact same thing. These 50% heterozygotes individuals will give themselves rise to half of individuals being heterozygotes and half of individuals being homozygotes. So that means what? That means that basically the 50% left of heterozygotes in the F1s will give half of the progeny being heterozygote. So we are only left in the F2 with 25% heterozygote individual. So that shows you that basically every generation, the proportion of heterozygotes will decrease by 2. Okay, and we're gonna end up after a very few generation actually with a purely homozygote population if we are purely selfing. So increase homozygosity across time, and guess what? As we discussed, that means that we're gonna have less efficient recombination and less efficient selection in selfing populations, and the same apply to populations that are experiencing a high inbreeding, okay? Because high breeding will exactly leads to the same situation with increased homozygosity. Which brings to another important idea. And this important idea is what we call the mutation load or genetic load, used a bit in the same manner. So what you have here is a distribution of mutations that range from being completely neutral to being completely lethal, and with all the range of possible deleterious effects you can imagine in between. And you have this in two populations, in outcrossing, non-inbred, so kind of, let's say, healthy population in terms of genetics, and another population that will either be inbred or selfing, which again, as I said, leads to more or less the same result. You can see a difference in the distribution of mutations between these two types of population. First of all, if you look at very highly deleterious mutations, you see that usually there is actually less of these very highly deleterious mutations in the selfing or inbred populations. And how can this be explained? Usually that these mutations are actually recessive. That means what? That means that in outcrossing population with high heterozygosity, these mutations are usually masked. So the selection cannot act on them simply because they are recessive, okay? So they are sort of invisible to selection. But when we look at selfing or inbred population, it's opposite. Homozygosity make these mutations visible for selection. And that means that individuals carrying these mutations will be purged out from the population. So maybe counterintuitively, for very highly deleterious mutations, selection can act more efficiently in selfing or inbred populations. But now let's look at the other side of the spectrum. So this time we are with mutations that are rather not so deleterious. They are slightly deleterious, I would call, which means that an individual carrying this mutation will not be counter-selected that much. And there we have the problems that we discussed before, which is that because of homozygosity, we're going to have high linkage disequilibrium between beneficial mutations and these slightly deleterious mutations. But because the balance is still kind of neutral or in favor of these beneficial mutations, these deleterious mutations, slightly deleterious mutations, will not be purged out. They will be each hiking these beneficial mutations. Okay? And on the long term, that will lead to an accumulation of these slightly deleterious mutations in the genome, which is exactly what we call mutation load or genetic load. And across time, it is predicted that the fitness of the overall population in a selfing or inbred will slowly decrease because of the accumulation of these slightly deleterious mutations. And that's just a summary graph, basically, which kind of summarizes everything I told you. So with the bottleneck, honor effect, selfing, polyandry, which then change the sex ratio. And as we discussed, the more imbalanced sex ratio, the more distorted, the smaller population size, effective population size we have. And of course, parthenogenesis is the exact same thing as selfing happening. And you can imagine clonal propagation, etc., etc. So with all of this, this will decrease the effective population size, it will decrease polymorphism, and together with like increasing homozygosity, and that will then decrease effective recombination, that will then, which is more or less the same as saying that there is increased linkage disequilibrium. And all this together will decrease the efficiency of selection, both positive and purifying selection. And because it decreases the efficiency of purifying selection, that will increase the genetic load in general. And on the other hand, because positive selection cannot act properly, then the adaptive potential of this population will decrease. Okay, now a quiz for you to summarize before we move on to the next step. I'm asking you now, according to the current paradigm, what drives most of the genetic diversity? Is it selection, is it stochasticity, or is it demographic events? So think about it, pause this video, and write down, you can comment this video in perusal, so just write down your answer there so that the others can see it, and then you can discuss with each other if you agree with each other or not. And if not, why, etc., and that will help you. That's what we call peer learning, which means that will help you a lot to learn if you have to debate, if you have to argue for your own answer, etc., etc. So for all the quiz we're gonna have in these videos, please write down, don't be ashamed, write down your answer there. I will not interfere, I will not tell you what is the correct answer. You have to discuss it with your classmates. So A, B, C. And it can be, you can choose several answers. Of course, it could be one, it could be two, it could be three. So again, what drives most of the genetic diversity? Next one. If we have a bottleneck, inbreeding, selfing, that will decrease what? Genetic diversity, the efficacy of selection, linkage disequilibrium, efficient recombination, homozygosity, effective population size, stochasticity on allele frequency changes, and what we call the genetic load. So again, there might be several answers that are correct here. Put them on as a comment on the description of perusal. Okay, let's move on to the next step. So now that we went through all possible events that can affect the population, now the next step is to measure them, to detect them. Because as I said before, we can talk about selection and search for selection. We first need to test the null hypothesis. So to test the null hypothesis, which are demographic or stochastic events, we need to be able to detect them. So how do we do that? First of all, we can simply measure very simple parameters. Typically, nucleotide diversity, that will tell us what? That will tell us about the genetic diversity of a population. A simple way to do it is to measure what we call pi. You probably have heard of it, and it's just simply measuring the average pairwise differences. So imagine that you have 4 individuals with their own sequence, and of course it's just singular sequencing, but the same applies to genome, to a whole genome, of course. So we can measure the difference of sequence between individual 1 and 2, between 2 and 3, 3 and 4, 1 and 4, etc., 2 and 4, and so on and so forth. And then we're gonna make an average of how many differences are found in these pairwise comparisons. That's pi. We can then do another thing, which is measuring heterozygosity. So we have several genotypes here, okay? So big A, small, like homozygotes, big A, homozygotes, small a, or heterozygotes. And you might remember that basically, no, that's something else, sorry. So this is just to tell you, okay, so we can measure the frequency of alleles, we can measure frequencies of genotypes, okay? So frequencies of big A is P, let's say, and frequency of small a is Q. And then we can measure the observed frequency of heterozygotes, so the observed heterozygosity, and here there are 4 individuals being heterozygote out of 7. So that gives us 0.57 of a frequency of heterozygotes. Now, with simply with Hardy-Weinberg equilibrium and Hardy-Weinberg expectations, we can measure what we call expected heterozygosity. And the way we do that is simply 1 minus the sum of all the allele frequencies to the square. So typically here we have 2 alleles and their frequencies that we say P and Q, and it means that we need to make P and Q to the square, and then we make a sum of all of this and we remove this from 1. So if we calculate it, you can calculate it yourself, you will actually find that the expected heterozygosity is 0.5. So of course here it's just a simple example that has no value, but you see that there is a deviation to a certain extent from the expected heterozygosity. So what does it mean? Well, it's pretty obvious that if you have a lower observed frequency of heterozygotes compared to what you expect, that suggests you that there is some selfing or inbreeding going on because we are losing heterozygotes. If we have more heterozygotes than expected, then of course it becomes a bit more tricky, but we can think of different things like whatever heterozygotes being more fit, for example, so they will have the tendency to be in higher frequencies in this population. Okay, and now, so there is another a bit more elaborate way to detect demographic events, and that's calculating what we call G-D. That's a bit tricky to explain, so and I'm not a population geneticist. So there is actually another video from a guy, Dr. Noor, that explained it very well. So when I saw that video, I thought I cannot explain it better than it does. So next video will be him explaining the G-D, and then we can simply go back to our lecture. So enjoy.